Hello, good morning, everyone. Welcome. Good morning or good afternoon, wherever you are in the world. I'll just share my screen here and we'll start in a few minutes. I see people are still joining. Um, there we go. So this session is being recorded. All of our webinars are being recorded. So if you need to access the recording, you can always go under flojo.com in under the webinar tab, and there, or simply type in Google recorded webinars Flojo, and you'll find all the list with the webinars, including this one a few days after. So I guess either on Friday or next week, the recording will be available to you. So you can go over it if you have more questions. Good morning, good morning. I see familiar people there. So we'll start in 1 minute. Just to let you know, if you have any questions at any time point, please do use the Q&A box or the text box, and I'll go through the questions while I'm presenting to you. Okay, so I see no further attendance, so I think we can start. Good morning, everyone. My name is Yannis Panetas. I'm a senior scientific advisor at Flojo Informatics, which is part of BD Biosciences. My email address you can find right here. It's ioannis.panetas@bd.com. In case you have any questions after the end of the webinar, I'm really happy to take anything related with your data analysis within Flojo, whether this is from imaging data or not. Yeah. So today we will be focusing on this webinar on analyzing data coming from the S8 FACSDiscover with Flojo and its plugins. Yeah, so this applies and will be applied also in data coming from the A8 or any cell view technology from BT. I'll show you the workflow of how we can analyze such data. So we'll be focusing on basically how we can handle those imaging data and bridge those with the flow parameters. Yeah, so as well as how we can exactly go back to the sorter with an optimized gating strategy, which is something we will use the HyperFinder algorithm for. Yeah? And once again, for the people that joined us now, please, if you have any questions, type your questions in the chat box, and I'm gonna regularly go through. Okay, so let's get started. So I like to start this webinar by going through the journey of dimensions we receive in a software like Flojo and how exactly we got there. Yeah, having a very difficult task to analyze such complex data and multiple dimensions. Yeah. So in the beginning of flow cytometry, of course, we had very few parameters to analyze. So we had a few instruments that had few lasers, few detectors. So we would maximum have 2 or 3 parameters in there to analyze, which is Of course, something you can still do today, and it's really, I never say to a user that it's bad to have on your research 2 or 3 colors. If it makes the job, if it does the science, it does it. Yeah. So we're happy for it. So in Flojo, it's quite easy, this task. Yeah. So if you're familiar with Flojo, you can place your gates, you can overlay populations, you can have a gating strategy, copy to the group, and export all statistics and graphs you might need. Of course, then with the advancement of cytometry, also with new instruments, spectral instruments, more precise, we can now identify many different subsets or immune cell subsets within our samples, so we can use more markers. Yeah, so now this is called multi-parameter flow or high parameter flow. And you can already see here the comparison of a low parameter and high parameter flow experiment, how different it is now in terms of, all the possible combination of parameters we might have and different subpopulations. Yeah. So in such a case, you might have heard already the high dimensional workflow we use. And this specifically in Flojo we can do with the use of different algorithms or plugins. Those could be either dimensionality reduction algorithms and clustering algorithms, as well as visualization tools. So this makes it all much more simplistic to extract all the information and to visualize our samples simultaneously in one plot. Yeah. And now what we're addressing today is even more dimensions. Yeah, and now we can go not only in high parameter flow, where we have all those flow parameters, but we can have more information about where is the location of this fluorescence, what is the shape of our cells and morphology, cell interactions, specific subsets that are with flow parameters the same, but with imaging parameters different. Yeah, so apart now from the flow parameters, we also have images recorded, and this is something you can do with the S8 FACSDiscover sorter. And in addition, we have imaging parameters. So those imaging parameters are basically mathematically calculated, and they describe still morphology. It's like a translation of the images into mathematical values. Yeah. So now the point here is, how can we analyze such a data set? Yeah, if we have more than 100 parameters in a flow sample, how can we analyze? And this is exactly our topic of today, and I'll show you the solution we have within Flojo, which is the BD CellView Lens plugin. What does this plugin does? Similar with the high parameter flow, we again have the target to visualize everything at once. Yeah, so we want to still perform our t-SNE, dimensionality reduction, clustering, all those algorithms, but we naturally want to see the images now, yeah, because for once we can now really go over the graphical window and see each one of the dots and see a picture for each one of the dots that we couldn't do in the past. So the BD CellView Lens is a plugin. I'll show you in the next slide where you can find it and download it for free. And I'll show you a workflow on how we can use that exact plugin in order to analyze such data. Okay, so that's our topic of today. So for installation, if you're familiar with Flojo plugins, those can be found under flojo.com. And if you go to Flojo Exchange, here you will find a whole list of algorithms we have, and you can download them. So there you will find the BD CellView Lens plugin. So please try. I know we constantly update those, but we update them for a reason. Yeah. So the BD CellView Lens is currently at the version 1.3.15. It's a software we always, find things in there. So try to keep updated. This is the one I also use here today, which was published 28th of October. Yeah. So to download, you need to drop in a folder of your choice or a folder named plugins. You can drop the jar file of the algorithm and then specify within Flojo under preferences and diagnostics. You need to specify where that folder is. Yeah, and after you restart Flojo, you will find under your workspace those plugins, and specifically the self-view lens plugin, but this applies, of course, for all the different algorithms we have. Yeah. So I'll show you now the workflow in the total workflow we're going to use here today. Yeah. So let's say you have the instrument. So I guess for you to be joining here, you possibly have or thinking of having the instrument. The instrument uses BD Fax Chorus. Yeah, so you will record your sample there and mix your samples. And then you will have an FCS file, as we typically have the flow cytometric standard file. And together you will have a CVW file. This is the like a zip file, a chorus wave format file, which is basically all the information for the images. Yeah? So here is a representative data size, just so you have an estimate. This is 400,000 events that we have together with a corresponding wave format file. This can be then unmixed as a next step. Extracted, not unmixed. Unmixing happens a bit earlier. This can be extracted with the image extractor, and then you will have a folder with TIFF files, which will be the actual images that we will add within the BD self-view lens plugin. So after you generate those files, we do have a solution to where you can store those in the BD Research Cloud, because you already see that we have massive data sets. So if you acquire 20 samples, then of course it's going to be different, and you might need a different solution to store those and directly have them accessed within Flojo. Yeah, so the Research Cloud is the solution we have. And once you have all those, yeah, the FCS files and the TIFF files, you can drag and drop your FCS files within Flojo, run the plugin. The plugin will then ask you for where exactly your TIFF files are located. You will give that input, and then you can start the analysis. After the analysis, our point of today would be to try to find a population, yeah, and try to simplify its gating strategy. We might find a cluster, we might find a specific morphology. We'll see a little bit later what exactly we'll be looking. So we might want to simplify in terms of, if you have 200 parameters, which are the most important ones to use in order to sort that population in the future. And this is where exactly the HyperFinder algorithm will help us trim all this list of parameters to find the most optimal gating strategy, which you can then even transfer back to Fax Chorus and sort that population or cluster you have identified. Yeah, so that's the whole workflow, the circle of workflow we're going to be using here today. I see a question there. It seems the screen is not okay. Maybe you can click on, yeah, there must be the screen. I also see I'm sharing the screen. Let us know. Maybe you can double click anywhere else. If you see a small icon of the presentation, double click there to expand the window. Okay, let's continue. Just to recap a little bit about previous webinars on assay data. We previously had a webinar. Of course, it was not European time, it was US time, so it wasn't really convenient for us to join live, but it was recorded. It was one webinar from my colleague Nicholas Love. So basically, we're not going to go through today about data management so much. We're going to focus more on the plugin and the imaging analysis. But I can suggest you this webinar if you want It's very fresh, and key takeaways from these webinars was basically how you can scale your data, how you can save your scaling, and store your data in BD Research Cloud and access them in Flowjo. So it's basically a workflow using the algorithms of BD CellView Lens within Flowjo. It's just to prepare, have consistent data within your acquisitions. Yeah, so if you want, again, go under recorded webinars in Flowjo and you can access that there. Okay. Okay, so for our workflow here, whenever we have the FCS files, the first step typically is to drag and drop the FCS files in Flowjo. So here we, for example, added a pre-sort DC sample. Then you need to go, after you have successfully installed the plugin, you need to go under the Workspace tab and select Plugins and run the BD CellView Lens plugins. Then, important is that the plugin, as the first window that appears, is the directory. It needs to find where exactly your images are stored. Yeah, so it will try to find it automatically. Sometimes it might not find automatically the folder with the TIFF files, so you need to input it by yourself. Most of the times it will find it. And this is here a recommended structure that you would have in the folder. Yeah, so you would have the FCS file, you see it here, and then you would have a folder that is named exactly as the FCS file with the images. Yeah, within that folder, you would have every 10,000 pictures you would have in a different folder. Yeah, so this is what this is here a correct folder structure that you would have after you export this CVW file. Yeah, and then of course the nice part is we can start analyzing. We can see the images in different kinds of filters or different anything different we apply on the visualization. I'll go through all those tools later when we go into the demo. So the first thing you're going to be seeing after you open the plugin, you're going to see the main window of the BD CellView Lens plugin. That consists of three main parts. Yeah, so you have on the left, you will always have the image wall. So the image wall is basically displaying each one of the events you have with the correct number here. You can also switch to from image from wall view. You can also switch to channel view to view each individual channel that was recorded. And you can scroll through the different pages of all the events you have in there. Yeah, you can add more rows, columns. We're going to see all that. The right-hand side, you will see a more high resolution, more zoomed in picture of the event that you selected on the left. And the same for the individual channels here below. You'll see all the individual channels. And finally, the ribbon down below is the image display ribbon, where really we can spend a lot of time there. I'll show you. It's really like a new software. It's really the most comprehensive plugin we have in terms of things you can do, but I'll show you the most important ones like report making, the interactive mouse over function, and many, many additional tools like image sets and so on. Okay, so of course, what I would do first when I drag and drop my samples, it would be to setting up the image filters. Yeah, so every microscopy experiment, or if you're already performing, you would know that the alpha and omega is to set up those. Yeah. So we do have the image filter button where you can adjust the threshold, you can eliminate the background, adjust the minimum and maximum threshold, you can add smoothing to each individual channel. And you can preview here on the right the overlay. Yeah, so the plugin will make an overlay of all the channels you have active here on the left, so you can activate and deactivate. You can also import or export the filter sets that you have so that you still, again, have consistency across your experiment. And you can select which channel here on the right, which channels are applied back to the workspace, so that, if you want to see only here, for example, the gold G staining, you can click on it and then apply filters to workspace to only indicate this in the wall view. Yeah? And you can add, of course, the naming of all your channels and the imaging channels you are using. Once we set those up, we will go to the next step, which is running the plugin on multiple levels. Yeah, that's of course what we want in the end. We want to place our gating strategy, yeah, and have few gates and run then simply the plugin in multiple levels. Yeah, once I run it, on my doublets or my live cells or a specific population, I can simply drag and drop this cell view lens node to any different population, and it will only show me the images of that specific population. Yeah, so we don't run it only once, yeah, but only only on the different levels we're using. Yeah. I see a question here in the chat box. Could you please mention how to export only the subset of images corresponding to a specific gate? Yeah, so export, you mean, I'm not sure if you mean export the actual TIFF files, or you mean export, maybe you mean either to visualize the specific gate, which I just explained right here. But in case you mean export, we also have an option I'll show you later. When after you run the plugin on a population, if you go to the tools tab, I think I'll show it to you. You can select their export PNG files. Yeah, so then you will, for only the selected population you have, you can export the PNG files, and then, depending on if you want to use them in other software or to visualize it differently, you can use those pictures. Yeah, I'll show it to you in a minute when I go live in Flowjo. Okay. All right, so we run the plugin in multiple. We run the plugin in multiple populations. So something else that it's really something that helps our discovery, and it's the best part of it actually, is the interactive mouse over function. Yeah, so this goes two ways. Yeah, so you can either open the graphical window that you have in Flowjo, you're familiar with it, and you can just go over the dots, yeah, over the events, and visualize the images of each event. Yeah, and that way, of course, this will help you have more accurate gates. For example, here you see I have a singlet gate and I see some doublets. Yeah, so I might then move that gate a little bit to the left to make sure that I don't have doublets in there. So you can imagine I can have now much more accurate gates, and I can find specific morphologies by using a two-dimensional plot. And that, of course, doesn't stop there, because you can imagine then we can have a T-snitter or a UMAP and even see the different islands of those embeddings and how its morphology corresponds to a different island. That goes two ways, because you can also click on the pictures on the image wall, and this will link back to exactly on the graphical window. Yeah, so if I start clicking here to different events, I will see back to my graphical window those where they live on this graphical window. Okay. Once we identify the population of interest, we can make a lot of batching within the layout editor. Yeah, we have some new functions in there I'll be showing you later. The easiest is you just click on the button of, so it's the photograph button, where you basically will transfer your image world and the high dimensional, the high resolution image in the layout editor, and you can export it later on. So you can do it multiple times for the multiple population as a report of what exactly is the morphology of each subset you identified. And then, of course, the one on the right, you might see here, it's a nice addition we have. There is a shortcut for it where you can select your population and you can visualize, if you have an image set that we're going to explain later, if you're creating a set of images or a specific cluster, you can visualize those images back onto your graphical window, and will respond like it will directly connect each image with a specific dot or specific event you selected there. Yeah, so you can really define nicely the Disney embedding or human embedding and so forth. Yeah, so that's something I'll be showing you later. Now, new stuff, we have the image sets. So only for this function, I would tell you that you really need to download the latest version of the plugin because it's there, where basically you can create a new population, let's say, or an image set, where you can click manually which images morphology you want to put in there. So you can click on a morphology that is interesting to you. It could be apoptotic cells, GFP positive cells, anything you want. You can click on those and add them as a new image set within the plugin. Yeah. So what can we do with that now? You might ask me, okay, so now I have specific morphology. So first of all, you can export it, as previously was asked in the Q&A box. You can export the PNG files. Then you can also create a new population out of it. So you can make a new population in your workspace that includes only the selected image sets and the images within. Yeah? So it will bring it back to your workspace. It will look like that. So here I selected 12 cells, and this will now give me a population under here, which is 12 cells and images pop1. And of course, then I can run a plugin on it, or I can use it for further downstream analysis. Then the nicest function that I like the most is the quick discovery of similar images. So let's say we have this image set of those doublets here, and we want to find in this full sample how many morphologies like that exist. Yeah. So we do have this nearest neighbor function, where basically you need to click on this button here, and it will identify the nearest neighbors, meaning what are the most similar cells that are having the same morphology as our image sets. Then you need to include which parameters you want to compare, because of course we need multiple parameters to compare to find how similar they are or how neighboring they are. So here I can use my flow parameters, I can use my imaging parameters. The more I use, of course, the better, because then I would really find only the ones that are the nearest neighbors. And after, I need to input also how many neighbors I want to find. Yes, in that case, I selected 50 neighbors, and then this will run very fast, and it will then show you a result, which will be a new population, which is called similar to image set 1, okay, with all the populations, all the sample, all the images that are similar to the population I selected. So it will really find directly in a very fast way. It will trim all the data and only find similar images to the morphology I have my interest on. Okay. And I can make as many image sets as I want within the workspace. Okay. Other functions for sorting, we do have the index sorting. So if you acquired your data or if you sort it on a plate, it will also generate, apart from the FCS files and the images, it will also create a CSV file where you have the well IDs, all the well ID information, and you can actually visualize a plate. You have this plate view where you can have like images of only the selected well you have, yeah, so that you know exactly what you sorted, and you can even further create the well populations back to the workspace. I have some data in my demo. I'll show you very quickly how you can actually look at those. I see, hi Tharsan, I see she asked a question, how much time to run the quick discovery? 5-10 minutes or 30 minutes, 1 hour? Well, it depends. So I haven't, if I'm honest, I haven't gone too far with it. I haven't gone to, I don't know, 100,000 events and look for 3,000 cells. What I can say, and I'm not sure if it's like proportionate, and you'll see it later in the demo, is finding 100 or 500 cells within 10,000. It takes seconds, not even minutes. It's really impressive how fast it is. Yeah, so I would, for quick discovery, I would say around 5 to 10 minutes if you push it, but you'll see later it's quite fast. No problem. And then let's say now we identified the population, we made an image set and found all the similar images to the one, the morphology we're interested. But we now don't know how to exactly sort that population, yeah, because we this step involves some manual selection of images. Yeah, when we created the image set, we selected morphologies manually. So there are two options. So now, in order to find the gating strategy for this population to sort it, you can either form like hundreds of two-dimensional plots where you see the, you know, where they land on each plot, or you can make a t-SNE, but you still don't know what would be the parameters that, you know, are expressed the most or the least, and how you can separate from the rest of the sample. And that's where HyperFinder comes in place, because it can really trim down just to the parameters that make sense and offer resolution in order to sort that cluster. So the way it works is you select your population of interest on the right-hand side. So here we have, for example, PDCs. And then you need to select which parameters you want to use for identification. Yeah. So it's not a problem if you use more parameters. So if you use all of them, it will of course take longer time to run. But if it finds no variance for some parameters, let's say the side scatter here or the light loss, they don't have any variance, it will exclude them automatically. It will say those are not helpful, I remove them, which is great because it will trim it down by itself. And then it will start first in the high dimensional space to create some gates and find what is the optimal gating strategy. It will adjust the gates in order to identify the highest yield and purity of that population. And you get a score in the end that defines, you know, how good is this ratio of purity and yield. Yeah? And this gating strategy is then transferred in your workspace. So you will find, depending how many gates you selected, you will find the gating strategy, Hi5 from HyperFinder. And those gates can then be transferred back to Chorus. So if you're familiar with Chorus, there is the button to upload, to import your workspace from Flowjet directly, and you can use those gates for sorting. Yeah, of course, keep in mind that if it's a different acquisition, you still might need to adjust a little bit the gates to fit in your sorting strategy. Okay. Okay, so that's the overview of what we're going to be seeing here today. I want to introduce you to the dataset before I go to the analysis part. So this dataset is HeLa cells developed with the EMBL in Germany, and specifically I want to thank Benoit Winkler from BD, from the training center in Belgium. I don't know, I think I saw you, Benoit, in the meeting. I really want to thank you because this is one really good data set that I can demo you the CellView Lens plugin. It consists of, it's a GFP cell line, and it has a 3-color staining. Yeah, so it has a surface staining, which is CD147 in PE. Okay, so we're going to be seeing the membrane staining. Then we have DRAG5 to visualize the nucleus. And then, of course, we have the GFP in Golgi. Yeah, so we will, you'll see this signal, intracellular signal, of course, but not as wide or diffuse signal as the rest of the parameters, imaging parameters we're having here. Yeah. And what we're going to be exploring is all the tools we said. So we will form a little gating strategy, 3 or 4 gates, and then we will run the plugin on different levels to visualize the images and go into discovery. Yeah. So demo time. I don't know if you have any other questions before we get started. Otherwise, we'll see everything in the software. Okay. So I'll quit here. I will just reorganize here my windows. Okay, there we are. That's my workspace. I added the FCS file, which is 3-color HeLa, it's called. First of all, just to, if you haven't used data from the S8, I will double click here just to show you what is the difficulty we're dealing with. And the difficulty we're dealing with is the parameter number. So you see all this parameter number is extensive. Yeah. So especially in that case, we need to use a strategy where we simplify this. And this is where, dimensionality reduction tools and clustering, and also the CellView Lens plugin is going to help us to identify the most important information in there. Okay. So how do we start? Well, I'm not going to start directly with a gating strategy, or maybe, yeah, we can place one gate just to don't have any debris in here. I'll place a gate around there. Okay. I will name it cells. Okay. And now, if you don't want to run the plugin to the whole population, you can also, of course, run it in. You can run it into the cell population. Yeah. I see. Sorry, I see another question here. You mentioned data can be transferred to the BD cloud and stored there. Then is the analysis running on your computer or on the BD cloud? The analysis is going to be running on your computer. So the BD Research Cloud will store your data and store. You can extract your CVW file there and then import it to Flowjet, but then you still, the computational power would be from your own computer and not the cloud. It's just for the storage at this point. Okay. All right, no problem. Right, we select our population, go under the workspace tab, select our plugins and select BD Cellview Lens. It's the first plugin here, it's alphabetically. So the first time you run it, you're not gonna see that. Actually, just to simulate, the first time that you run the plugin, you will see something like that. This, okay? It will ask you for where are your images, yeah? So you need to select where your images are located here, or you can access them, as we said before, through the BD Research Cloud. I'll use this function later for the index sorting, but you can click here and have access to your Research Cloud and load your CVW file in there. Now, the folder, so for me, you can see here, I have a folder, Cellview Lens, I have my FCS file here, and I have the folder with images here. Yeah, so you see I have 10,000 pictures approximately. So Flojo will, the plugin will find it very fast if you have, based on the naming, it can find this folder very fast. If not, you can just press on the folder here and find exactly, where is this folder, and it's that folder right here. Yeah, and then once you press OK, well, before you press OK, you should see that you have this green check. So it means it found exactly the images we're looking at. If you see an X, it means it hasn't found yet the images. And in addition, if you have an index sort file, CSV file, you need to click here and load this CSV file to visualize, in its well, where exactly are your images. Yeah, so then you press here, OK, and then it will open the main plugin window. Okay, so here you see the BD Cellview Lens plugin window, where, as we described earlier, you see on the left the image wall. Okay, so you can switch between wall view and channel view if you want. Yeah, so here we are at the channel, we see each channel individually, and Flojo constructs also an overlay, of all the different channels here. Yeah. I typically prefer the wall view at the beginning so that I can adjust separately each channel to be seen nicely. Sarsana asked a very good question in the chat, which is, if we merge concatenate FCS files, can we still use the Cellview Lens? Well, yes and no. So if you concatenate the traditional way your files, if you have multiple samples and you go to File, Concatenate, then it's gonna be a no. Yeah, we cannot do that yet because the event number is lost. But I will show you, so I can show you directly now maybe. So we do have, if you go under the tools, okay, how to explain? So apart from the functions you have here below, it's important to know that in the BD Cellview Lens plugin, you have also the tabs to use. If you're on a Windows, this could be integrated. If you're on a Mac, you see those View, Tools, and Pref tabs. Yeah. So once you have run your plugin into multiple levels, let's say I run, here and here. Yeah, those two in those two population, I run the plugin. And I multi-select those. Let's say let's imagine it's from different samples. Yeah, you have different samples and you multi-selected those two. And then you go under the Tools tab and select the option Export Selected Cellview Lens Population. What it will actually do, okay, it will create a new concatenated file, FCS, where you can actually visualize the images of each separate population we merge together. Yeah, that's the only option. If you do it traditionally, you cannot do that. But yeah, in that case, I cannot really show you that here. I have only one sample but that's the process. You multi-select your BD cell view lens nodes in your workspace from the different population. And then you go under the Tools tab and select Export Selected Cell View Population. Yeah, it's working. All right. So within the window here, you can select, it's one of the different events you're seeing, and this will update here the view on the right. You can, on the bottom left, you can add more rows or more columns if you want to visualize more. And you can, of course, scroll through the next pages and, next set of events here. Yeah. Something I really like to do a lot is if you want to sort your populations based on the expression or the value of each one of one of the parameters, you can use the ascending or descending here. So for example, if we wanted to see here, okay, show me first which events have the highest GFP. Yeah, so I can do something like, okay, I'll press here maximum intensity GFP. Okay, and then instead of ascending, of course, I will press descending. Yeah, so those events here, maybe I can use the channel view. Okay, those events here, you see that they have high GFP in here. Yeah, if I had it in ascending, you see no signal for GFP. And the same I can do for any of the parameters, whether those are flow or imaging. So for example, eccentricity. Yeah, so now here we see the highest eccentricity. So those, of course, are doublets and debris here. Okay, and lowest eccentricity I can do as well to visualize singlets. Yeah, so that's a very nice way to trim your events very, very fast. In terms of reports, if you're like in this view, you can press the button here below the photograph, and it will make a report directly into your layout editor here. If you hold the Shift button and press the photograph, it will also transfer the magnified view of the event you have selected. Yeah. And then I'll show you a bit later how you can make it tracking each event where exactly on the plot. Yeah, just a moment. Now, Flojo will load one of your filter sets, presets. So here it's very important because if you don't see what exactly you were seeing on the instrument, it's something you can change here. Yeah, so if you click on here, you can see I have different filter sets. Yeah, so basically which channel has which color, smoothing, etc. So we have here no colors, auto color smooth, auto colors without smooth, and etc. Yeah, so I can always select at first glance which one works the best. So I would select auto color smooth in that case. But then we, of course, want to make more changes here. Yeah, I have too many active channels. I don't need all those. It's just colorful. I will modify this as my next step. Yeah. How do we do that? I'll click here on the image filter tool to modify those. And here is the main window where we can modify its individual channel. Yeah. How do we start? Well, we start by selecting each one of the channels here, deciding on whether we need them in or out, and then modifying each one. Yeah. So light loss. light loss in microscopy or bright field, it's a really nice channel to have. I would keep it always in. It's to your preference, of course, but we will need to modify this. It's a little bit dark here. Yeah. What I can do here is I can use either my minimum or a minimum threshold, but it's full here, or use the gamma. Yeah, so with the gamma, really you can remove a lot of background to make everything much brighter. You can also add here smoothing, yeah, if you want to make it a bit more blurred, yeah, so that it's not so distorted. That at this point, I think I'm satisfied. I can move to the next channel. I have three main channels I want. It's those imaging here. So I think I can skip the forward scatter and the side scatter here. Yeah, so I don't need to keep them in. You see at the bottom, let me magnify here. You also have magnifying so that you can see better the different channels. You can magnify here and see the overlay that the plugin creates of the different channels. Again, with double click, you activate or deactivate the channel. I will deactivate those, and then I'll move to my next one. This channel is my Golgi. I can also type it here on the right-hand side. I can give it a color, okay, maybe a bit brighter green, and I will name this Golgi GFP. Okay. And here I will try to remove a bit the background. You see we have some background here. I can remove it. Yeah, you do have this option, which is also new. You have the option remove background, and this removes all the background that is outside of the region of interest, and it will remove it and only keep it inside the cell. Yeah, so I typically use that to remove that from the overlay. Yeah. If you want, you can, after you remove here the background, you can select the option to sharpen the signal. In that case, it's actually good because it's a few pixels that are Golgi GFP in there, so I would need to make it a bit more visible. Right. Then move to the next channel. Okay, here is the CD147. You can change the colors of each channel here. Yeah, so you can click on the color, you'll get the color palette. I will use an orange, nice color here. And then again, I will make some modifications. Okay, so I will play around with the gamma. That's my first option always. Okay, I will try to remove background and then adjust smoothing if I need to. Okay, I'll do something like that. Yeah. Right, and then I'll go to my drag 5. I can also load my name of my file here, drag 5. And then again, I will adjust my gamma, smoothing. Okay. And the nice thing about it is whenever you make modification, you can also press here, show overlay. Okay, and this will show you directly the overlay of all the different channels. We do have different kinds of overlays. Just add a bit of smoothing here. We do have different overlays. The one I'm using here is the blended, so it blends each channel equally with each other. We do have the transparent as well, where basically you use one as the scaffold and then you add another layer on top of it. So ideally in that case, you would start with 100% for the first layer, and then you would go lower and lower to the next layer. And then finally, we also have the solid overlay. It's not really my favorite, it's more distorted. So typically I start always with a blended overlay and see what I get there. Now, in terms of, you see also another button here, find best threshold. What is this? So this is basically the masking. Yeah, what region do we have as the region of interest that is displayed here? Yeah, I typically also select that, and you see, yeah, you see directly that it found a better masking of what is exactly where is your cell located and which till which pixel it expands. If you want to adjust that, you can do that by holding the Shift button. And so you see here, hold Shift key and select on the imaging channel. So what this will do, it will now activate here this overlay, and you see what exactly region you include in here. And so if I do that, for example, now I selected a much larger area. Yeah. So this is how you can adjust, what exactly is your region of interest. Yeah. But I typically, trust best to find the threshold from the algorithm here. Okay. Now, if you have if you have a image filter that you're satisfied, you can now save that. Yeah, so you have those buttons here, and I can say save the current filter set, okay, and I can name it, for example, filter set webinar. Okay, and then of course, if I run the same experiment, I can then load that XML file that it creates and have the same filters with the naming and everything. Okay, I know it was a lot here to talk about, but it's really important to set those accurately. Yeah. Then, if I want to apply these changes, okay, I need to select Apply Filters to Workspace. Okay, so this will apply everything to my workspace, and you can see that in the image wall. If you want to display one channel, well, I can select one channel here on those buttons and show only one channel. So now I only show the drag 5. Now I only show the CD147, and now I only show the GFP. Yeah. So the overlay in that case would be ideal for us. Okay, and it updates everything. So now I think I'll close here the image filters, and then I will move to the next topic. Yeah, don't know if there's any questions. I just saw something happening. Yeah, so there's a question. From your experience of self-view parameters, are you able to comment on which imaging analysis parameters you would recommend to look at cytoplasm to nucleus ratios to measure cell morphology changes and disease progressions, perhaps also cellular metabolism? Well, that's a hard one. Well, it's really, I cannot comment on that and say which imaging parameters it would be. Hopefully, I will show you how you can trim those down, and when you need to algorithmically define which parameters are playing the most important. I will show you this. I cannot answer that, unfortunately, because we see now data coming from phytoplankton to cancer samples, infection with tuberculosis. There is so many experiments that I really cannot. I expect that different samples would have different parameter importance. Yeah. In our case here, for example, what we're going to be looking is some cells with close interaction. So it might be that eccentricity plays the most role. If you are seeing, larger cells or stimulated cells change morphology, it might be diffusivity or other parameters play a role. It's really not an easy question to answer there, but I think it always will depend on, it will depend on the samples and types of samples you have. Okay. Maybe I'll show you some insight on how you can find those. Okay, let's move on. We do have some additional tools here below where you can, first of all, select random images. Okay, so if you want to see any image we have in there, you can also click on regions here. So it shows you the regions that used as a mask here. Edges, so it forms here an edge around the cell. Connection, so this is important if you want to see doublets. I don't have any doublets here, but you see how it connects. Here we have a doublet, and together it will calculate the distance from one cell to the other and draw a line here. Yeah. You can activate all those from the bottom right. Now, the next part is gating. Okay, so you can click on the interactive mouse plot here. Okay, so this button, and this will open our main window of graphical window where you now can go over the dots and see the pictures. Yeah. So as I go here, you see I have more singlets. When I go further, you see all those doublets. Now, I can also change the parameters here. So something I could do is to select here my eccentricity, light loss, and my radial moment, light loss. Do it like that. Okay, so again, here we would see more eccentricity would show us more doublets, less it would show us the singlets. So I can maybe place a gate here, name it singlets, and another gate here to name it doublet. And you can even, while you're drawing the gate, you can even inspect around and have a much more accurate gate. Yeah, that's really nice. Okay, so that's how you can use the interactive window to display your images while you're gating. Now, I can close that. I can also close this window. Okay, and now I can run multiple times the plugin anywhere. So if I want to see my doublets, I can drag and drop here on my doublets, singlets, and open those windows. So if I do that, okay, you'll see here I'm having my singlets. Something I could do is I can still change that gate and it will refresh everything. So if I now see, okay, I have doublets here, well, move that gate around, right? So I can move that gate around to avoid any doublets in there, yeah? So it's all refreshing live, yeah? Whenever you change a gate and you run previously the plugin on it, it will update the images, okay? Right. I see chat box. Okay, don't see anything. Okay. Right. Okay. And the next step of the gating strategy here, maybe going from singlets, would be to see Golgi, for example, max intensity Golgi versus diffusivity Golgi. Okay. And this is something we could see. So here you see we have Golgi high cells. Yeah, so I could click on here. Again, click on my interactive window. This will open my gate and I can see here possibly my Golgi high cells and I can place a gate on it. Okay. Now, can do something like that. If I click on the images here, just to show you this as well, if you click on any image here, this will also point back to the plot and where exactly your cell is located. So you don't see it very well because it's red here, but if I use any kind of different visualization, you'll see it much easier. Yeah, so these are Golgi high here, okay, etc., etc. Okay. Right. Let's go into the next topic here, which is the next topic is going to be the, as I said before, the image sets. Yeah. So just before we do the image sets, let's make some dimensionality reduction. Yeah, just to have it in there. I can perform dimensionality reduction on my whole cell population here by simply selecting it. I can run t-SNE or UMAP. I can simply run a t-SNE here and select which parameters I want. Now, now you have to make a choice. Do you want all of the parameters? You can do all. Yeah, so I can select all the parameters. It's going to offer me much greater resolution of the islands. Yeah, I can also select my fluorescence parameters and my imaging. Yeah, in that case, I will just select my imaging parameters here and run just a basic t-SNE here, just to have a structure of the cells. Yeah, in an embedded space. Now here I don't have a lot of events. I have almost 9,000 events, and t-SNE is already done. Yeah, so I see some separate islands here. I could explore those. Yeah, display some large dots. I see, for example, here I have high eccentricity. Okay, so it might be here a region of doublets. I can also look at max intensity of Golgi. Okay, well, this population here might have some interest for us, but we also have some high max Golgi here as well, yeah, which would be doublets. To get, and this I can explore also live interactive in the plugin. Now, for the next step, I can also perform clustering. So I can again select my population, go to Phenograph or any other algorithm. And again, I will select my fluorescent parameter, my imaging parameters, perform a clustering just to have a structure, different clusters for this cell population. And then we're gonna go next. Keep it in your mind, and we're gonna use that later in the analysis as well. Yeah? Phenograph will take slightly more time, but it should be done soon. And then we'll make an image set soon. Sorry, I did some pre-work just to show you that those tools also work with the SA data here. And what I'm gonna do next is I will create a new image set of cells that are in close contact. I'm not gonna use the term interaction. I don't know if they're really interacting, but I'm just gonna create a new image set. We're gonna manually select phenotypes that are in close contact, okay, cells that are like doublets but in close contact. And we're going to use the nearest neighbors function in order to find 100 cells, 100 events that are looking like that. Yeah. And then we're going to see where this lies on the t-SNE plot and see how we can explore that with HyperFinder further on. Yeah. So Phenograph finished. So here, the new version of Phenograph, if you have it, shows you also a very nice hierarchical clustering together with the signature of all the parameters you selected. And Phenograph found here 12 clusters. Yeah, so you see we have clusters 1 to 12. You can always do things like overlaying those. Yeah, so you can select here on your t-SNE 1 and t-SNE 2. Just keep that for later. And then I can drag and drop, of course, my clusters in here and visualize them on my t-SNE. Yeah. So, and that's very nice because you see, how nicely they still separate the populations into different clusters based on the imaging derived parameters. Yeah, that's what we wanted. And imagine if you add also the flow parameters on top, you will have even better separation in there. Yeah. Let's keep that in mind and move to the image sets. Now, image sets. Just a question first. Can you use the imaging parameters in R? How does one integrate with the fluorescent parameters, FCS plus TIFF file, using the plugin extract integrated FCS and then load in R? I think you should be able to use those parameters in R. It's like flow parameters, they just scaled a bit differently. So what you could do is to export those. Yeah, so you can, don't forget, you can, if you have an FCS file and you want to maybe open it in R, you can go to File, Export, Populations, and then you can select here Export. Okay, and instead of an FCS file, you can use a CSV file. So you will have a list of all the values, whether those are fluorescent or imaging, and you can certainly use them in R. Yeah. Okay. Right. Now we go to the image sets. So I will open here my cell population, and then the image sets you will find on the bottom right corner. Yeah, so you will be seeing in the newest version of the plugin this show image sets. Okay, so you see here I have 0 image sets. I don't have anything. But what I will do is I will create a new image set. So on the left, you're seeing all the image wall you have, and on the right, we will create a new image set. Let's make here with the plus button a new image set. I will call it close contact cells. Yeah, like that. Press OK. And then you need to unlock it, yeah, because it's locked initially. And now I can start clicking on images here, and this will transfer it on the right. Yeah. So now what we want to find is cells in close contact. So what I will do to quickly find those is to basically use the sort ascending or descending order. Yeah. So if I now put, for example, my eccentricity for P, the surface, and then unclick it, so it will start showing me first, yeah, the highest eccentricity for the surface staining. So here are debris. I'm not going to select any of those. I will go, now I see triplets. I will still go further and much further till I start finding those close contact cells. And I start seeing them here. Yeah, so I see one here. Okay, we could say that those are in contact. Let's move, this one maybe as well. Okay, I will just select a couple of them manually. So of course, the more you select, the better, certainly, because it has more to compare with. Okay, this one looks contact. So of course, this is a bit of a manual part. Yeah, I need to manually click on the morphology if I'm interested. Yeah, here I see some close contact here as well. I'll make just a few more. So here I see I'm in a hotspot of eccentricity where all of them pop up. This one, yeah, so here I see a lot. Yeah, so it could be eccentricity that is very important for this exact population, but it remains to be found. Yeah, and I think I'm gonna call it here. Yeah, maybe those two as well. All right, so now I made my image set. Yeah, so this includes those events that I manually selected, and I want to transfer it into my workspace? Well, quite easy. You click here on the population button, and this will bring it to your workspace. Okay. So if I go to my workspace, you will see I have, I selected manually 33 cells, and those are my image set, and then those are all the rest of the cells, yeah, that I don't want. Now, if I want to find the similar neighbors, I click on the button here, where I can select which parameters I want to compare to find the nearest neighbors. I will select everything here. Yeah, so I can select all of the different imaging parameters and also fluorescent parameters to find, let's make 150 events that look like my image set. Yeah, so I'll just press here accept. So you'll see how fast it is. It's impressively fast. Okay, amazing. And the morphologies look great. Yeah, it's. I don't see any doublets. I see cells in close contact in here. Yeah, if I see different morphologies that are not really the ones that I selected in my image set, it's because I needed to pay more attention. If I start to see doublets, it's because maybe I selected one that was not really a close contact cell, but a doublet. Yeah. So now it found 150 images in here, and then those images I can bring again back to my workspace. Yeah. So then I will press here create population, and it will make a new population here, which is called similar to close contact, and I have 150 cells in here. What I can do more is run the plugin in there, like just drag and drop the plugin, open this, and then I have my cells in here. Yeah? All those doublets. How can I go? Yeah, so the question is, I can see some triplets in there. How can I remove them? Yeah, so that's my bad. It could be more pure, but how you can remove them is you can go back to the image set in here and then unlock, select your image set, unlock it, and then you can remove those. Yeah, so you could start removing some of those. Yeah. So, and also to a question that was asked before, no problem. To the question that was asked before, you can export the PNG files. So if you have here your image set, you can go under the Tools tab and send here, create PNG files, yeah, for this population, which is nice to do. Okay, and then I can even, what I can do is I can overlay this population I found, yeah, on my Disney. That's where it gets interesting. I can drag and drop this population here and place it onto my Disney. Well, maybe I can do a separate Disney because it looks that I see it better. Yeah, I'll put my Disney 1 and Disney 2. Okay. And I can overlay this population. Okay, and you see this population lies over here. Yeah, so this would be exactly this will be exactly where my cluster would, we can see which cluster this could be, right? That's not difficult. I can overlay my clusters here, and I suppose that this is my cluster. So my population is right here, and it should be phenograph cluster number 4. Yeah, this exactly is my cluster. This red one, no, I'll make it like a different color. Uh, yeah, any color, green. Um, so this is might be a cluster that I have interest to identify later on. Um, so now we can move to how we can identify what this expresses. Yeah. So the traditional way of doing that is, you could do a heat map in here. Yeah. So you can do a heat map and see, for example, which parameters are expressed here, or use this phenograph um, clustering to see what your cluster expresses and design a gating strategy. But we're not going to do that here. We're going to use HyperFinder to identify this cluster number. I already forgot. It was cluster number 4, I believe. Yes, it was cluster number 4. Okay. Well, you can also run HyperFinder just on the similar to close contact. That should be no problem. And I will click on my full FCS sample, workspace, and then under the population tab, you will see HyperFinder. Yeah, I'll click here. So on the right-hand side, you need to define the population. So I can select either my cluster or my similar to close contact cell population 1. Um, and then um, we will select the parameters that we want to compare. Yeah, so here I will select my imaging parameters. Yeah, so that next time I could have a gating strategy only with the imaging to identify the specific population. You can select what is the maximum number of gates. I tend to reduce it. I don't want to have 8 gates because the algorithm sometimes I notice that it tries to force a little bit to find to add more gates in there. And then I will select the polytope gate finding algorithm for studying this population in the high dimensional space. It's like a polygon in the high dimensional space. So I'll press here OK. The algorithm will start running. And this is the part I mentioned before. It tells you that some of the parameters doesn't have any variance in the target population. And this is here the parameter size, yeah, size light loss. So I don't need to include that. Even better for me, I can include all the rest and remove that parameter uh, to find that population. So that's a way to trim which parameters also play a role and can have distinguishing power. So I'll press here, OK. Tarsana asks another difficult question. If we don't know the number of cells we want for nearest neighbors, will the algorithm try to fit the number we give? Yeah, I believe so. So I think if I would press, well, it could also be the case in my data set here that I forced it a bit. Previously, I tried it with 100 and it was okay. With 150, yeah. So it will, if you overdo it, yeah, it will find more dissimilar events. So there's no way at the moment to find the best number there. But if you select 2,000 nearest neighbors, it could be one neighbor too far away, right? It will still calculate this and put it in there. So yeah, that's a difficulty we're having. But to quality check that, maybe you can, start with a number and see if you have impurities in there that you decrease significantly. No problem. Right, so the algorithm still runs here. You see, I have a lot of dimensions in there to fit in, but yeah, let's see what we'll get out of it to identify those close contact cells. Are there any questions in the meantime while this is running? Check the Q&A. It's empty here. Okay, so almost done. Few more dimensions, and then let's see what we're gonna get out of there. Because the traditional way of doing that process would involve a lot of two-dimensional plots, right? So to find that cluster, that specific population and what exactly it's, how it should be gated, it would be difficult. Yeah, I would need maybe, I could also use Cluster Explorer to see all the different parameters and see which ones are the most differential. That I could gate with, or yeah, or use the Disney and start to make different heat maps and see what this specific island expresses in that case. Yeah, so here it can give you insights if you don't want to transfer the whole gating strategy to Chorus, you can still get some insights of which parameters are quite different than the rest of the events within that sample. Yeah. We do have a few minutes more, 6 minutes more, but I think it will be done by then. Maybe still select few less, but yeah, I overdid it a little bit, but this will be done soon. It just takes all different combinations of parameters, puts them on the 2D plot to identify the optimal gating strategy in there. Okay. Yeah. And yeah, so needless to say that you can, when you perform dimensionality reduction, clustering, and all those tools, you can always still drag and drop the plugin node into a separate cluster and see what which images we have in there. You can further export the images. There is really a lot you can do with those tools, and this is where those tools get much more significance. Yeah, when you have such a long list of parameters, um, it really makes sense to use dimensionality reduction more than ever in that case. Yeah, because we can visualize everything at once. It's much easier to interpret and identify your population. So HyperFinder here is finished. It will use a different combination of gates. So I see that the most important parameter here is eccentricity and diffusivity for identifying this population. So it formed different kinds of gates. And then those HyperFinder gates you will find here below. Yeah, so you can even open those up. Yeah, so you can go back and see the whole gating strategy it formed to identify this specific population. And you can run it multiple times into different population on the ones exported from the similar or nearest neighbor function within the plugin clusters or anything really you want. So maybe the results here could be better, but in that case, I could still run HyperFinder again. And so there's no problem. Can select few of my parameters just to show you a quick example. Correlation, radial moment, diffusivity, etc., and select cluster number 4. Yeah, so I believe here we'll get a better result. Certainly faster, yeah, because I used much less parameters, and rerun it for every dimension. Let's see if we'll get a much more better result here. Yeah. So that's much better result. Yeah. So you'll see the result here in the F measure. So this is the ratio between. Ideally, we want 1 there. Yeah. So here I see with radial moment P, eccentricity P, this was the surface staining, and then with size P versus radial moment P, and then diffusivity P. Yeah, versus diffusivity goals. It identified that specific cluster. And this gating strategy, again, can be transferred. And we simplified one cluster into 3 different gates. And this can be applied to any kind of phenotype you have in your experiment. And you can even try something you could try is if you have a large panel, like 8 colors, for example, you could try HyperFinder with imaging derived and see if you can get the population that you used 8 antibodies on to see if you can gate it only with imaging features. Yeah, that would be also a very interesting way of using HyperFinder. Okay. So that's the last part I wanted to show you. And I want to thank you very much all for attending. I know I went a bit over time, but there was a lot to show you in there. We're certainly going to have more webinars on CellView Lens. Thank you very much. And yeah, if you have any other questions, let me know. Otherwise, see you in the next webinar. Thank you. Thank you, Benoit. Ah, you were there. Thank you all. Thank you.